Hello, my name is Deward Lee Whitlow, and I'll be presenting the article Ultra-High Throughput Clinical Proteomics Reveals Classifiers of COVID-19 Infection by Christoph Messner et al. It was originally published on July 22nd, 2020 in Cell Systems, and here you can see the digital object identifier. Here you can see the points we'll be going over in this presentation. We'll talk about the overall goals of the paper, the necessary background information, the methods the authors used, their main findings, and any limitations or strengths that I saw in the paper itself. The paper had two main goals. The first was to create a standardized ultra-high throughput clinical platform for serum and plasma proteomics. This platform would be inexpensive, provide high throughput. It would handle up to 180 samples a day. It would reduce batch effects, and it would enable high precision quantification. The second goal was then to apply the platform to COVID-19 patients. They wanted to identify potential biomarkers that define disease severity. They wanted to perform this task quickly, and they wanted to identify therapeutic targets. All right, so necessary background information. The first thing is understanding bottom-up proteomics. Bottom-up proteomics involves protein digestion through enzymes. Typically, trypsin is what's used, and that's actually what is used in this paper. The peptides are analyzed by liquid chromatography and mass spectrometry, and then the data is mapped back to known proteins. It's also helpful to understand data independent acquisition, or DIA. It's a technique used in mass spectrometry. All the fragments are scanned in this technique versus data-dependent acquisition, DDA, where they cherry pick. They just find the most expressive proteins and image those. The paper often refers to ISO 13485, and what that is an industry standard for medical devices. These devices are things that are used for design, production, and installation. And what it is it certifies quality control. It's also important to understand batch effects. These are variations in data caused by differences in sample collecting, handling, and instrumentation that happen on different runs or days. And what they can do is they can make variations look like meaningful biological results when all they are is just a different way, different handling styles or different variations in the handling. This paper also makes use of Generation Scotland. It is a tissue bank. It's a family-based cohort of approximately 25,000 individuals in 7,000 family groups from across Scotland, aged between 18 and 98. These are people who volunteered to donate tissue samples that are then used for a wide range within the medical research. The paper itself used the epidemiological study Smith et al. 2013. And then we have COVID-19. And I mentioned this because of the time frame that this paper came out. It was July 2020, which was the height of the pandemic. There was no vaccine. The researchers were still trying to get a handle on what this virus was all about. So this paper actually gives us a kind of like a sneak view of how scientists were handling this emerging pandemic. And then something to understand about it was the WHO severity grades. They went 1 through 6, and just each grade represented a different severity of the virus. So moving on to the methods used. So we'll start with the platform itself. The authors actually included a workflow so you could actually see the process of their platform and what all is involved. In essence, there's 4 parts to the workflow, starting with receipt and storage, going on to preparation, data acquisition, and finally data processing. We're going to start with receipt and storage. So in receipt and storage, samples are collected using standard operating procedures. They're received and stored at -80 degrees Celsius. Stock solution plates are preset for the whole project, and that's part of what they are, what they're presenting as making this a more streamlined production is that they're able to get all their samples that they need and have it already stored and ready to go to be plugged in at different parts in the workflow. Those solution plates include trypsin, four batches each. They have their denaturation and reduction solution, which also comes in 4 batches, and then the iodoacetamine, and that's 1 batch. All right, moving on to sample preparation. Again, in the previous step, they had prepared stock solutions so they can use throughout. And here you can see where they're plugged in at various stages of their preparation. So here we'll take a closer look at that sample preparation method. Samples are aliquoted into 96 well plates. A denaturation and reduction solution is added to them. What that does is it unfolds the proteins and makes it kind of linear so that they can then be digested. That solution is then centrifuged and then mixed and incubated for 60 minutes at 30 degrees Celsius. 5 microliters of iodoacetamide is then added, and what that is doing is making sure that those bonds that you've kind of broken apart do not reform. They're incubated in the dark because it is a light sensitive process. They're incubated in the dark for 30 minutes at 23 degrees Celsius, and then they're diluted with a buffer. They're then transferred to the trypsin plate, and that's what cleaves the proteins for microspectrometry analysis. This whole process takes about an hour and a half, and it is done through robotics. All right, and then that sample plate is then incubated at 37 degrees for 17 hours, typically overnight. After that, the cleaning process begins, and that takes approximately 2 hours. Formic acid is added to it because we're trying to get to a solid phase extraction. There's a minute of centrifugation, and then a liquid handler used to pipette the liquids into the material to make 4 96 well plates. They're conditioned with methanol and washed twice. They're equilibrated twice, and then they're centrifuged again. They're then completely dried on a vacuum concentrator and redissolved into solution, and then they're stored at -80 degrees until it's time for data acquisition. And here we see the data acquisition step. In it, high flow liquid chromatography is used. It's used at 5-minute gradient lengths, which substantially increases sample turnover and reduces cost while increasing stability and precision. So instead of going super short or much longer to get a better picture, they've kind of found like a median range. And then they use the swath program and swath acquisition to get the data. And so now we're at the data processing portion. A closer look at the data processing shows that they use the raw swath data that they got in acquisition, and they apply it using a data independent acquisition software. They basically import a project independent spectral library that acts as a reference map. They then use that as a map for the peptides that they have imaged. The library is then refined using the data set. They reanalyze everything, make any batch corrections, run it through a protein quantification software, and then lastly they make the biological interpretations. So the authors then wanted to demonstrate the practical applications of their platform. They first demonstrated this platform using Generation Scotland. They generated proteomes from undepleted serum samples derived from 199 random individuals from the study. There's also a large number of commercial plasma and serum that were added in as well as quality controls. They showed that their platform yielded exceptional retention time stability across the whole experiment. The number of peptides quantified lower than other MS workflows, but it was much faster while still covering what they felt was the most typical plasma proteins. The authors then turned their attention to COVID-19. They wanted to use their platform to analyze patients that had been hospitalized with COVID-19. Samples were collected from 31 patients in the hospital in Berlin between March 1st and March 26th. These were patients that, these are initial patients that are being hospitalized from the COVID-19 pandemic. They used their platform to identify potential biomarkers, and they compared them with the World Health Organization severity grading. 17 further patients at the same hospital were then used as validation of those biomarkers. All right, so what they found. The authors were first able to demonstrate a high throughput clinical platform. This platform is scalable, it's automated, it's quick. They're able to do 96 well plates in about 3.5 hours. They then turned that platform towards COVID-19, and they were able to identify 27 potential biomarkers. Those markers were able to differentiate severity in the World Health Organization rankings, and they were able to highlight the role of complement factors including coagulation system, inflammation modulars, pro-inflammatory signaling upstream and downstream of interleukin IL-6. For example, you can see there was a decrease in expression levels of APOA1, which is a modulator of inflammation. And then in the coagulation system, there's upregulation in the coagulation factors. All right, and so next we're going to talk about the limitations of the paper the way I see it. And several of these the authors freely admitted themselves, and they all really revolve around the COVID-19 portion of the paper. The first is the sample size. It's a small sample size that they're pulling from. The way those samples were collected, these are just the nurses and the doctors in the hospital, so there could be variation in the way they were collected and handled. Also, there's a time component. These samples were collected at the initial stage of the pandemic, and there's no real continuous sampling, so it's only this snapshot of this one initial point. And finally, there's no independent validation, so they used the same data set to refine itself, and there's no way to independently validate what they were doing. And now for the strengths. We'll start with the platform itself. The authors used open source software, so they're able to show reproducibility. Others are able to follow along what they did and reproduce what they were doing. They used an automated process. They had robotic arms that were doing the pipetting of the samples and the solutions. What that did is made sure everything was uniform and actually created a more rapid analysis because they were able to cut the manpower time down of each processing time to about 3.5 hours. Everything also became more cost effective, and then lastly, they were able to reduce batch effects by presetting their solutions before they started handling the samples themselves. Moving on to the COVID portion of the paper, they were able to identify 27 biomarkers identified with COVID severity. They were able to provide insight into protein function, and they provided a rapid response to emerging pandemics. So while they applied this to COVID-19, they basically showed that in the future, the next thing that comes along, there is a way to respond to it quickly. So that's my presentation. Hope you enjoyed. If you have any questions, please leave them in the comments. Thank you very much.